Welcome to the interactive video presentation on microbiological evaluation of ready-to-eat foods. In this video, you will be watching a demonstration on how to microbiologically evaluate a ready-to-eat food to decide whether it is fit for human consumption or not. You will learn how to use microbiological guidelines in making decision on fit for human consumption of a ready-to-eat food. Ready-to-eat foods include food that are prepared in advance and therefore will not be cooked or reheated before consumption. These include salads, cooked meats, smoked fish, desserts, sandwiches, cheese, a combination of various food components and food that you have cooked in advance to be served cold. Ready-to-eat foods are associated with the occurrence of foodborne illness cases and outbreaks, raising concerns about their safety. Some of the common foodborne pathogens associated with refrigerated ready-to-eat foods include Listeria monocytogenes, pathogenic Escherichia coli, Bacillus cereus, Staphylococcus aureus, and Salmonella species, just to mention a few. To ensure consumer safety and in preventing foodborne illness, such food products must meet set minimum microbiological criteria before it is sold on the market for human consumption. Such microbiological criteria are set by national regulations as legal requirement and compliance is mandatory. Guidelines are, however, developed by food safety regulatory agencies including public health authorities in respective countries, in support of the regulations on microbiological criteria. Ready-to-eat foods must therefore be tested and must meet these set criteria. Criteria are also applied for bacteria that indicate possible poor hygiene and/or substandard practices. Failure to meet the criteria alone is not an offense, but the specific corrective action must be carried out to comply fully with the regulation. Microbiological criteria are intended to assist with validating and verifying HACCP-based food safety management systems. In this demonstration, we will be following laboratory protocols to analyze a ready-to-eat food, using various selective and differential media, as well as non-selective media. We will then look at how to interpret the results generated from such analysis to decide whether the food is safe for human consumption. The following materials are required: weighing balance, vortex mixer, various selective and differential media plates. A non-selective media plate count agar, used for total viable count or aerobic colony count. 9 ml Ringer's solutions in dilution bottles. 250 ml Ringer's solution for diluting food sample. Salmonella 1 broth for salmonella enrichment. Listeria 1 broth for listeria enrichment. Stomacher bags. Stomacher bag opener. Stomacher machine. Sterile spatula or spoon. Sterile spreaders. Bunsen burner. 1000 microliter micropipette with corresponding pipette tips. 100 microliter micropipette with corresponding pipette tips. Now let's start by sampling the food in which we want to detect and identify microorganisms using the selective and differential media. We will be sampling and microbiologically evaluating barbecue chicken pasta as our food sample. Before sampling the food, you need to label all the dilution bottles with corresponding dilutions levels. The agar plates are also labeled on the agar side with dilutions levels and ID to identify your samples. In this demonstration, only the dilution levels are shown as labels. The food is sampled using the following procedure. 1. Aseptically place a stomacher bag into the stomacher bag holder, place on the balance and tear the balance. 2. Sterilize the spatula or spoon and take a sample of at least 10 to 20 grams of the food sample. Make sure sample is taken in a manner to reduce the possibility of handling contamination. We have taken 12 grams of food sample. 3. Add Ringer's solution to the bag. Make a 1 in 10 dilution. Example, for 10 gram sample, add 90 gram Ringer's solution. In this demonstration, we have weighed 12 grams of food sample. To determine the amount of Ringer's solution to add to the food sample to get the required dilution, multiply 12 by 9. This gives us 108. That means the total weight will be 12 grams plus 108 grams, which gives us 120 grams. This is the same as 12 multiplied by 10. So simply add the Ringer's solution to the food to give a final weight of approximately 120 grams. (4) Stomach for 30 seconds to 1 minute in the stomacher machine. This becomes your starting dilution of 10 to the power minus 1. The stomacher machine is made of 2 metal paddles that beat against the sample in the bag, homogenizing it and releasing microorganisms into solution. Where small numbers of bacteria are expected, or a possibility of the presence of fastidious organisms, an enrichment process may be required. This enrichment media contains nutrients to encourage microbial growth to detectable numbers. Samples tested for Salmonella species, for example, are incubated for 24 hours in enrichment broth, such as Salmonella 1 broth, prior to inoculation onto selective media. After incubation of the enrichment broth, a loopful is then streaked onto the selective media, which is incubated at required temperature and time before observation. In this demonstration, we will use the selective and differential media used in Week 3 interactive video presentation. These include McConkey broth, eosin methylene blue, Baird Parker agar, mannitol salt agar, rose Bengal chloramphenicol agar, malt extract agar, Brilliance salmonella agar, Brilliance bacillus cereus agar, Pseudomonas selective agar and chromogenic listeria agar. We will use plate count agar for total viable count. Other names for total viable count are aerobic colony count, total plate count, total aerobic colony count. For the detection of Salmonella and Listeria, guidelines require that 25 grams of food is tested instead of 10 grams. The food sample is transferred into a stomacher bag and 225 ml of enrichment broth is added. This is then stomached and incubated overnight before streaking onto respective selective media. In the next step, we have to perform a serial dilution using the stomach sample. Remember, this is our first dilution of 10 to the power minus 1. Set the bag with sample on the bench. Open it carefully, ensuring no extraneous contamination occur to the inside. Using the 1000 microliter micropipette, aseptically transfer 1 milliliter of the homogenized sample from the stomacher bag into the first dilution bottle. This gives us a dilution level of 10 to the power minus 2. Mix very well using the vortex mixer. Next, open the cap and flame the neck of the bottle. Aseptically pipette 1 milliliter or 1000 microliter of culture from the first dilution bottle into the second dilution bottle, using a fresh pipette tip. This gives us a dilution level of 10 to the power minus 3. Replace the cap of the second dilution bottle and mix very well using the vortex mixer. Open the cap and flame the neck of the bottle. Using a fresh tip, transfer 1 milliliter or 1000 microliter from second dilution bottle to the third dilution bottle. This gives us a dilution level of 10-4. Replace cap, flaming and mixing as before. Repeat for rest of the series. Remember to always flame the neck of the bottle after mixing and opening the cap. Always use a fresh pipette tip to transfer the sample. The highest dilution in this demonstration is 10-7. The respective dilutions are now ready to be plated onto selective and differential media. As we have seen previously, there are two options of plating the diluted samples. The first option is to plate starting from the least diluted sample. In this case the least diluted sample is 10-1. This is the sample in the stomacher bag, which is also our first dilution. In this demonstration, we will however, plate starting from the second dilution 10-2. For this option, pipette tips must be changed to a fresh one after each sample transfer. Similarly, a new spreader must be used for spreading the sample on each agar surface. Using the 100 microliter micropipette, aseptically transfer 100 microliter or 0.1 milliliter of respective dilution onto corresponding agar plate, starting from the least diluted sample. Do this for all the selective and differential media as well as the non-selective media. In the second option, plating starts from the more diluted sample. In this case 10-7. For this option, the same pipette tip can be used to transfer the sample from respective dilutions to corresponding agar plates. Similarly, the same spreader can be used for spreading the sample on the agar surface, starting from the more diluted sample. Using the 100 microliter micropipette, aseptically transfer 100 microliter or 0.1 milliliter of respective dilution onto corresponding agar plate, starting from the highest diluted sample. Do this for all the selective and differential media as well as the non-selective media. Using the serial dilution method, allows for the enumeration or counting of the colonies and therefore estimating the number of bacteria in the food sample. Only colonies showing the characteristic growth are counted. For presence or absence test, that is detection test, the streak plate method is used. In this case, only the sample in the stomacher bag is required. There is no need to prepare a serial dilution. Note, using a streak plate method does not allow for enumeration of bacteria. To prepare a streak plate, refer to the interactive video presentation on gram staining and microscopy. After plating, the plates are incubated at respective temperatures and time as shown in the table. The incubated plates are now ready to be observed and enumerated. Using the knowledge gained from the previous session on the use of selective and differential media in detection and identification of microorganism, observe the plate for growth and morphology of colonies. Select only plates that are positive for respective organisms. For E. coli, you should have positive reaction for both McConkey broth and eosin methylene blue. For Staphylococcus aureus, you should have positive reaction for both Baird-Parker agar and mannitol salt agar. For yeast and mold, you should have positive reaction for both rose Bengal chloramphenicol agar and malt extract agar. Observe the other plates for positive or negative reaction in accordance with the tested organism. Various confirmatory tests can be carried out on isolated colonies from the various selective and differential media, to confirm the identity of the organisms. Refer to the practical handbook for examples of such confirmatory test. The next step is to select appropriate dilution levels, with colonies between 30 to 300, and count the colonies using a colony counter. Refer to the interactive video session on aseptic techniques. Now calculate microbial numbers in the standard form using the formula above. Do this for both selective and non-selective media. You can then convert the final count to the log format. Now that we have generated our results, we will use the guidelines for assessing the microbiological safety of ready-to-eat foods placed on the market from the Public Health England, formerly Health Protection Agency, to interpret our results. The first result to look at is the total viable count or aerobic colony count, which is found in Table 5 in the guideline as shown. First, select the food category for the sample tested. Now compare the results from the analysis to the count presented in the table corresponding to your food sample. In this demonstration, our food sample falls into the food category 2, that is foods cooked immediately prior to sale or consumption. If the result from our analysis is less than 10 to the power 3 colony forming unit per gram, the food is said to be satisfactory. Therefore, no action is required. If the results fall between 10 to the power 3 and less than 10 to the power 5 colony forming unit per gram, the food is said to be within the borderline. This means neither satisfactory nor unsatisfactory. We therefore need to investigate the source of the food, including supplier, producer, or retailer, and the stage of shelf life, before determining action. If other samples from the same source are also of borderline quality, further investigation may be appropriate. If the result is above 10 to the power 5 colony forming unit per gram, the food is unsatisfactory. We need to carry out investigation for reasons for high count. The result of the total viable count is also used to estimate shelf life and incipient spoilage of food. For instance, in the case of meat products, incipient spoilage is said to occur at a total viable count from 10 to the power 7. For foodborne pathogens, there are two approaches to result interpretations. In the first approach, pathogens that require 25 gram of food sample for analysis are considered, example, Salmonella, Campylobacter, Listeria, pathogenic E. coli, etc. These require a simple presence or absence test in a 25 gram of the sample. Data for comparison are shown in Table 1 of the guidelines. In this case, detection or presence of the organism in 25 gram of the food sample renders the food unsatisfactory, due to high microbiological risk and potential injurious to health. The food is therefore unfit for human consumption. Non-detection or absence of the organism in 25 gram of the food sample means the result is satisfactory and the food is fit for human consumption. Under certain circumstances, Listeria monocytogenes detection is carried out in 25 gram food sample by enrichment. For high-risk foods capable of supporting the growth of Listeria monocytogenes, such as some soft ripened cheeses, sliced meats, smoked fish, and pâtés. In the second approach, pathogens that require 1 gram of sample are considered, example, Bacillus cereus, Staphylococcus aureus, Clostridium perfringens, and in some cases, Listeria monocytogenes, etc. Data for comparison is shown in Table 2 of the guidelines. Using Bacillus cereus as an example, a count of less than 10 to the power 3 colony-forming unit per gram of the food sample is considered satisfactory, meaning the food is of low microbiological risk and therefore safe for human consumption. A count between 10 to the power 3 and less than 10 to the power 5 colony-forming unit per gram is considered borderline, meaning the food is of a moderate microbiological risk. In this case, it is advisable to consider taking investigative samples of food, raw food components and the food preparation environment. A count of above 10 to the power 5 colony-forming unit per gram of the food sample is considered unsatisfactory, meaning the food is of high microbiological risk and therefore not fit for human consumption. The food has the potential to be injurious to health. The next to consider is the guidance on the interpretation of results for hygiene indicator organisms in ready-to-eat foods. This include the family of Enterobacteriaceae, non-pathogenic E. coli, Listeria species, not Listeria monocytogenes, etc. Using E. coli as an example, a count of less than 20 colony-forming unit per gram of the food sample is considered satisfactory. A count between 20 and less than 10 to the power 2 colony-forming unit per gram is considered borderline. This indicates a possible evidence of poor hygiene due to undercooking, or cross-contamination from raw food, especially meat, food handlers, or food contact surfaces, as well as poor temperature and time control. Repeated or widespread detection in several foods or areas of the food production environment suggests an increased food safety risk. A count of above 10 to the power 2 colony-forming unit per gram of the food sample is considered unsatisfactory. This indicates poor hygiene due to undercooking, or cross-contamination from raw food, especially meat, food handlers, or food contact surfaces, as well as poor temperature and time control. The detection of E. coli can signify a risk that fecal pathogens are present. Results should be interpreted in conjunction with test results from other microbiological parameters. It is always advisable to consider results of specific organism in conjunction with others to help make informed decision. To conclude this session, consider the following questions related to microbiological guidelines and ready-to-eat food evaluation. You will have to do additional reading or research in answering them. 1. Give one reason each why the following are important to the food microbiologist. Escherichia coli, Salmonella typhimurium, Pseudomonas aeruginosa, Bacillus cereus, Campylobacter jejuni. 2. Explain why McConkey broth changes color from purple to yellow with the generation of gas in the presence of E. coli. You have now reached the end of this demonstration. You may watch it again when needed.